Hello, my name is BshC, and I'm going to talk you through research that I've been working on this past semester. And my project has looked at an enzyme called BshC, and I've looked to better characterize its structure using small angle X-ray scattering. So to start us off as to why this research is relevant, it goes back to a molecule called bacillothiol. Bacillothiol is a low molecular weight thiol present in some gram-positive organisms, and this can include pathogens such as Bacillus anthracis or Staphylococcus aureus. It has a couple of functions within the cell. Two primary functions it has are to protect against oxidative agents and to help detoxify the organism. Along the lines of detoxification, this can include contributing to antibiotic resistance of the organism. One specific example of this is resistance to the drug fosfomycin. This drug is commonly prescribed for urinary tract infections. And what can happen is there's an enzyme called FosB, which can catalyze the addition of bacillothiol to the epoxide ring of fosfomycin. And this can break open the drug. And now this drug is no longer effective against the organism. Now, organisms that have been mutated so that they are unable to produce bacillothiol have shown increased susceptibility to antibiotics and other stresses. And so our lab is looking to better characterize the enzymes that make bacillothiol so that this information, might be useful in the future to design inhibitors of these enzymes. Then the organisms would no longer be able to produce bacillothiol. And then these once resistant organisms would once again be affected by these antibiotics. There are three enzymes that work to produce bacillothiol. The first is BshA, which takes the substrates UDP N-acetylglucosamine and malate to produce N-acetylglucosamine malate. The second enzyme, BshB, is a deacetylase, and it produces glucosaminomalate. And the third enzyme, BshC, this is the enzyme that I study. It is a cysteine ligase, and it then produces bacillothiol. Unfortunately, the activity of BshC has not been observed in vitro. However, a couple of years ago, Cook Lab determined the crystal structure of B subtilis BshC. Now, in this crystal structure, BshC was observed to be a homodimer, and it had this overall kind of dumbbell-looking shape to it. And they also observed that the location of dimerization appeared to be between the coiled-coiled domains of the two subunits. So because you can't really study the activity of BshC, we look to further explore its structure and answer some more questions about that. And so there were really two questions that we wanted to ask. One was that, does BshC truly form a dimer in solution? While crystal structures usually depict what is truly going on in kind of biologically relevant settings, this isn't always the case. Sometimes when these crystals form, when their crystal packing occurs, it can kind of force certain conformations on the protein. And sometimes that's not actually what you would observe in solution. And so we wanted to see, does it actually dimerize in solution? And then the second question we had was, is this coiled-coiled domain, is this truly necessary for dimerization of BshC? And so to answer these two questions, we use size exclusion chromatography, small angle X-ray scattering. So let me break down what exactly that means. Size exclusion chromatography is a separation technique that separates molecules based on their size. And so with this, larger molecules will elute more quickly. And this is kind of an added purification step because when you do these measurements, you want your sample to be as pure as possible, or a monodispersed solution, so only one thing in the solution. And so then that leads into the small angle X-ray scattering. And so with this, you would have your very, very pure sample, and you would subject this to an X-ray source. And from this, you would collect small angle diffraction data. Now, this diffraction data provides very low resolution data about whatever you're measuring, so in this case, BshC. You would not be able to tell the specific coordinates of atoms, or you can't really visualize residues, but you can tell kind of overall what a protein or molecule is looking like. So in the case of BshC, we would be able to see if it looks like this dumbbell in solution. We would be able to tell if it looks like that. Now, all of these small angle X-ray scattering measurements, these were all collected at the BioCat facility at Argonne National Laboratory. And we used 4 samples for these measurements. These first 3 shown up here, these are all wild-type BshCs from 3 different organisms: Bacillus subtilis, Staphylococcus aureus, and Deinococcus radiodurans. And then this last BshC, this is a coiled domain truncation BshC. And so what that means is this BshC has had all of the residues in this coiled domain. These are all removed. And so when we obtain the purified protein, it will no longer have this domain present. All of these BshCs were expressed in competent E. coli and purified. And then, as I mentioned, we did small angle X-ray scattering measurements on them. So now let's move on to some of the actual data that we obtained from them. So this data, all of these plots up here, these are for the B. subtilis wild type BshC. And so I'm going to walk you through each of these plots. This first one up here, this is a size exclusion chromatogram. So on the y-axis we have intensity, and this is kind of a measure of time. And the important thing to note here is we're really only seeing one large peak, and this corresponds to the BshC. And so what this is saying is that the sample that we sent off to Argonne was very, very pure already, and it even helped purify it a little bit more. You can see a very, very small peak here. And so when we actually did the measurements, we can separate out BshC from even that little bit of impurity. The second plot down here, this is the scattering profile, and this is actually kind of really the data that you're getting from the small angle X-ray scattering measurements. So this scattering profile is the intensity from the center of the detector as a function of Q, or kind of distance away from the center of the detector. So these blue dots here, these are all experimental data points. And then the program has fit in red, that's kind of the line of best fit running through these data points. And so using this scattering profile, this is how we can get information about what BshC looks like. So then these 2 plots over here, these are both mathematical manipulations of the scattering profile data. So this first plot, this is a Guinea A plot, and this is the natural log of the intensity as a function of Q. And over here on the x-axis, we have Q squared. And with the Guinea A plot, what we're really looking for is linearity. And we do indeed see quite good linearity with this B. subtilis wild type BshC. Linearity is indicative that no protein aggregation is occurring, that our concentration of the sample was appropriate, and also that the solution was monodispersed. Again, that our solution was very pure. So the fact that we see linearity is very good, and that tells us that the data that we're getting from this is going to be of a good quality. And then the last plot down here, this is the plot that's really kind of telling us what's going on with the overall structure of BshC. This is a pair distance distribution function plot. So what that means is it's the distribution of a pair of points within the protein. And on the y-axis here, this is intensity, and this is r. And now this is actually distances in real space. This is in angstroms. And this r here, this actually gives us quantitative information about the structure of BshC. So the first thing to notice with this is where this line is returning to 0. And it's doing that at about 180 to 190 angstroms. And this returning to 0 actually corresponds with the maximum distance of BshC. Then the second thing to take away from this plot is the observance of these two peaks here. And now those two peaks, that actually corresponds with a dumbbell shape in solution. And so what this plot is really telling us is that wild type BshC is indeed forming a dimer in solution. It has a dumbbell shape. So that was really exciting to see. It kind of confirmed our hypothesis. So if we come down here, we can see some numbers associated with these measurements. These first two columns are associated with the Guinea plot, the first being an r², the second being a radius of gyration. And then these two columns are molecular weights that the program calculated using two different methods. So that's why the numbers vary so greatly. Now, the important thing to look at with the molecular weights is how much higher the wild-type BshC's molecular weights are versus this truncated BshC. Now, BshC has a molecular weight of kind of around 60 kilodaltons. And so even though these two methods don't agree on one molecular weight, it's kind of indicating that there are two subunits of these BshCs existing together, or it's forming a dimer. And then if we go down here to the truncated BshC, it's significantly lower, which is more indicative that it's existing as a monomer in solution. And that is indeed what we observed. So this down here, this is the pair distance distribution function plot of the truncated BshC. So again, we have intensity on the y-axis and the kind of distance in angstroms down here on the x. So down here, we're seeing this line return to 0 around kind of 75 angstroms, indicating that 75 angstroms is the maximum length of this truncated BshC. And then notice how different this shape is from the wild-type BshC up here. It only has one peak, and it kind of looks like a bell curve. And this shape to this plot, that is indicative of kind of a more spherical shape in solution. And so this is consistent with this truncated BshC only existing as a monomer in solution. And so this small angle X-ray scattering data, this really confirmed that our hypothesis was correct. The wild-type BshC will form a dimer in solution, and this is saying that the coiled-coiled domain is truly necessary for BshC to dimerize. Now, using this small angle X-ray scattering data, we can also do kind of an additional analysis, and that is using the Fox server. So this server allows for crystallographic structures to be compared with the scattering data, and it kind of compares how well they agree with each other. And so we used 3 structures, and they were all based on the B subtilis BshC crystal structure that was previously determined. So we could have a structure of a BshC monomer, so just one of these guys, BshC dimer, so this whole thing. And then we had a truncated BshC structure. So this is where we removed the residues in this crystal structure in that coiled-coiled domain, which would kind of provide a very reasonable approximation of what we believe that truncated domain would look like. And so down here, this is kind of some of the data that we got from these. So this first column is describing what structure was used. So in this case, this first row is the BshC dimer. So it'd be this big guy over here. This is the scattering profile that was used. So this is saying the scattering profile of the wild type BshC. So what we observed up here, we saw formed a dimer. And this is the chi-squared value. Now with the chi-squared value, the lower the number, the better the agreement between the structure and the scattering profile. And so what we saw is that for the BshC dimer structure, the wild type BshC, we get very good agreement with that. And then also with our truncated BshC structure and the scattering profile of the truncated BshC, we also see very good agreement. And up here, that's what it's depicting. So this is the wild type BshC structure with the wild type scattering data, the truncated structure with the truncated scattering data. And so that red line is the structure as the program kind of fits it into the experimental data that we obtained. And so we saw a very good agreement with that. Then if we look at these two rows, so this is the structure of a BshC monomer. So it'd be like just this guy over here in cyan with the wild type BshC data, which we saw existing as a dimer, and a very high chi-squared. So it's saying that no, this monomer of BshC does not fit very well with that wild type scattering data. And then this one, the BshC dimer, so both of these subunits compared to the truncated BshC scattering data fits very, very poorly. So really confirming, no, this truncated BshC scattering profile is not looking like a dimer in solution. Now, then that kind of brings us to this last row down here. So BshC Dynamics 3 is referring to kind of an initial dynamic simulation done by Dr. Mary Carpin and Ian Green, an initial dynamic simulation of BshC. And this simulation was done in the gas phase. And this Dynamics 3 is the structural snapshot at 45 picoseconds into the simulation. So when this particular structure was compared with the scattering data of the wild type BshC, we actually saw an even lower chi-squared value than the crystal structure. And that is what's displayed over here. So this is that structure at 45 picoseconds into the simulation fitted to the wild type BshC scattering data. Now, because this chi-squared is even lower, that indicated that that structure may be a better fit to the data than even the crystal structure. So to compare kind of what the differences are with this simulated structure versus the crystal structure, that's what's kind of shown over here. So in blue, that is the B subtilis BshC crystal structure, and in orange is that simulated structure. And so what we can see is that the simulated structure appears to be a more relaxed conformation of BshC. It's kind of increased a little bit in length. It might have opened up a little bit. And so we'd need to do further simulations of BshC to see if there's a better conformation that fits with this data. But as of right now, that structure appeared to fit the scattering data the best. So to summarize what we learned from this project, we confirmed that the wild-type BshC does indeed form a dimer in solution, and that the coiled-coiled domain of BshC that really is necessary for dimerization, and it will not dimerize if it does not have this domain. Using the Fox server, we demonstrated that the crystal structure of BshC and our truncated BshC structure, these both matched the scattering data very well. And we also observed that an initial simulated dynamics structure of BshC, we found one that actually produced the greatest fit to the scattering data out of all the structures that we compared. This structural information may be useful in the future to design and develop inhibitors of BshC, which would result in organisms that are unable to produce bacillothiols. This lack of bacillothiols may make them more susceptible to antibiotics. Thank you very much for listening to my presentation.